Hello students, welcome to LEELA'S tutorial. So today we'll discuss about conservation of germplasm. Right? What do you mean by germplasm? It is nothing but the genetic resource or gene pool, or you can say the genetic structure of that particular plant species. So why we have to conserve this germplasm? In order to protect the endangered plant species and also the plant species which have the commercially importance, right? So here when you see the conservation of the germplasm, how you define the germplasm? Germplasm is a genetic material of an individual plant which can be or which may be transferred from one generation to the another generation by asexual method or by somatic method, right? So here let us see what are the different types of conservation or conservative methods. So first it is the ex situ method and other one is the in situ method. Ex situ, what do you mean by ex situ? It is outside the site. That means outside the natural habitat of that plant you are going to cultivate them, right? So let us see that. In situ means within the natural habitat of that plant species. So first let us see what is the ex situ conservation methods. In the ex situ conservation method, it is again classified into three types. First one is field gene banks, right? The second one is the conventional method and the third one is in vitro method. So here in the first case, it is the field gene bank where you whatever the plants you have to or you want to conserve them, you are going to cultivate them, right? So that is called as plant conservation. So here the plant conservation can be done in two ways. The first one is botanical gardens. So here in the botanical gardens, the particular plant species will be grown and these trees or the plants are given the botanical name plates, right? So that is the botanical gardens. Otherwise you can conserve them in arboreta. What are arboreta? These are the areas where you are going to culture or you are going to grow the trees and plant species for research or educational purpose, right? Then here you have the another method, conventional type. So in this conventional, it is also referred as traditional method. So under traditional method, you have the orthodox seeds. That means here you are going to store the seeds. You are going to store the seeds by drying them, right? So when you dry them, you are going to preserve them for longer time period, right? That method is called as orthodox seeds, which is a traditional method. Then coming to the in vitro method. In vitro, that means outside the body, outside the plant body. That means here you are not culturing them naturally. You are culturing the plants in plant tissue culture lab, right? So here under this you have recalcitrant seeds. So here this is quite opposite of your orthodox seeds. Here these seeds are dried and they can be conserved for longer time. Whereas recalcitrant seeds cannot be dried, right? They are in viable condition and they are they survive only for the short time. They can be preserved only for the short time. Is that clear? So here under that recalcitrant seeds, how you can conserve the germplasm? By using clones, DNA or pollen. Okay, so normally we have studied our plant tissue culture techniques, right? That is the same thing here. Let us see. So here you have the tissue culture method. Another one is cryopreservation method and other one is slow growth technique. So here in vitro, that means outside the plant body, you are going to conserve the plant genetic material. So first thing is either you go for your tissue culture technique where you are going to select the explant and culture them, right? So all the techniques we have seen, the anther culture, meristem culture, right? Embryogenesis, androgenesis, cell cultures, right? So you are going to take any part of that plant species and you are going to culture them within a small area in the lab conditions. That is your tissue culture. And the second method of conservation of germplasm is cryopreservation. So here, in case of cryopreservation, the germplasm can be stored, right, for much more longer time. Here the temperature is very less, where we are making use of liquid nitrogen, right. The liquid nitrogen fumes consists of -196, or it is maintained at -196 degrees centigrade. So the explant or the germplasm you will be storing in this liquid nitrogen tanks. Okay. And you have different types of cryopreservation techniques, and what is the method, we will discuss that in next class in detail, right. So next, going to the another one, it is the slow growth process. So you, in order to conserve the germplasm, either you go on cultivating them continuously in tissue culture lab by subculturing process, or you can store them for the longer time, right. Otherwise, allow the germplasm to divide very slowly, or grow the germplasm with slow growth rate. So under that, you have low temperature storage, right. So when there is a low temperature, all the enzymes will become inactive. So what happens? The plant or the cell metabolic activity will be reduced, because of which the growth rate will be slowed down. Then next one is growth retardation. Either by means of chemical method or any other substance, you can slow down or you can stop the growth of that particular plant species or the germplasm, which you have to conserve. The next one is the minimal media. Here you are providing a minimum amount of nutrients to the germplasm. In that case also, you can have the slow growth. Then last one is low oxygen concentration. So when you are decreasing the oxygen, obviously the growth rate will be slowed down. Is that clear? So coming to the next process, see, now this is everything comes under the ex situ method, where ex situ method is classified into 3 types: field gene banks, conventional method, in vitro method, right. Again, under field gene banks you have plant conservation, whereas under conventional you have orthodox seeds, and under in vitro you have recalcitrant seeds, which is again classified into 3 types, that is tissue culture, cryopreservation and slow growth. Is that clear? So this one we'll discuss in detail in next class. Now let's move to the in situ type. So again, the name in situ itself represents that it is within the site. Right? Within the site means the plants are grown within their natural habitat. Okay. So now in situ conservation of germplasm is again classified into 2 types. First one is natural habitat and second one is in farms. So first one, under natural habitat you have national parks. So here in the national parks, it is the national government which is allotting certain portion or certain area in which these plants which has to be conserved will be grown, right, under the protective environment, and it is maintained in its own natural habitat. Whereas in case of next one, the wildlife sanctuaries, here the plants are grown in their natural habitat, right. Apart from that, the protection is given, right. The surrounding invaders, there must not be, they will be maintaining in such a way that these plant species are not invaded or harvested, or they are not grazed by any other animals and all. So they will be giving a complete protection within their natural habitat. Then third one is the biosphere reserve. So here in the biosphere reserve, again they are going to conserve these plants and they are going to conserve the biodiversity. Okay. The next one is the genetic reserves. So here in the genetic reserve, the genetic pool, whatever you have to maintain, you are going to see that the range of their population is maintained in that particular habitat. Is it clear? Next, going to the in farms. That means here again the natural habitat is maintained in the farms, either by farmers' field, that is agricultural crops, or the agricultural fields where the farmers will be cultivating naturally, or by traditional method. Next one is the home gardens. Here in home gardens also, where the social and economic life is combined, right? So either in the families or the home gardens, the required plants are cultivated. And the last one is the tribal areas. Again, it is a natural habitat where their tradition or any traditional crops or the medicinal crops, whatever they are cultivating, they are given the special attention. Is that clear? So here by these in situ and ex situ method, you can conserve the germplasm. Is that clear? If you have any doubts, put it in comments. And in next class, we'll discuss about the different types of cryopreservation and what are the methods involved.